Welcome to another video for biology. In this biology, we're going to be looking at the process of turning RNA into proteins, or otherwise known as protein synthesis or translation. So as we look at the process of protein synthesis, we are going to be looking at the three main players, the main role players in this process. You have mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. These are all RNA molecules, okay? They're just different forms of them. They have different shapes, different jobs to do. So let's take a look first at rRNA. This is the RNA that you're already familiar with, actually, because it's the same RNA that leaves the nucleus that's been transcribed from DNA. This is also called messenger RNA. Okay, so just think of it as a messenger in terms of this molecule is carrying the information that originates from DNA. Okay, so our messenger RNA is the first molecule in the process of making proteins. So the second is tRNA, or transfer RNA. Transfer RNA is made out of three components. The first one is the amino acid. Okay, this amino acid is called aspartate. And then the second is the tRNA, the transfer RNA. This is the molecule that carries the amino acid and the anticodon. This anticodon, this section of the tRNA has a very specific 3 nucleic base code for an individual amino acid. So for example, aspartate has the code GAU. That's the anticodon. Okay? And alanine has GCA. So if you look going back to aspartate, you'll notice that as I change the amino acid, the anticodon, the code changes as well. Okay, and then glycine. Glycine has a code of GGA, and you'll notice for every single one of these amino acids, they actually have multiple codes, 3 base codes. So glycine, for example, has GGA, but it also has GGG. Okay. Now, if we take a look at valine, again, valine, another amino acid, having a different anticodon. Where do we come up with, or how do we know which amino acid is being coded for? Well, if you look in your textbook, you'll see a chart that's similar to this one that's going to help you in identifying which amino acid goes with which anticodon. So keep in mind that as we take a closer look at this, you actually have 20 amino acids, okay? So the tRNA goes together with mRNA in this way, okay? Now this is an incomplete picture, but I just want you to understand how tRNA and mRNA are related. So I'm looking at my mRNA right now. You can basically break down the mRNA into sections of 3, triplets, okay? 3 nucleic base. Each one of these 3 codes is the codon for the anticodon that goes with the tRNA, okay? So for example, we'll take a look at this first one. Okay, we look at our chart. This chart is based off of the codons. So if I have an AUA, that means the first base is A. So that means it could be any one of these amino acids shaded in blue. And if I look at my second base, okay, so you see my second base here is a U. So that means it's going to narrow down all of these bases to this particular column, okay? And then I take a look at my third base. My third base is an A, so then that narrows it down to AUA, which is isoleucine, okay? So that's the particular amino acid, or tRNA. tRNA will bring in this particular amino acid, isoleucine. All right, let's take a look at the second codon so we can get another example of how this process works. Okay, so the codon is UUU. Okay, so that particular code codes for lysine. Okay, so that is mRNA and tRNA. That's how these go. So mRNA has the codon, tRNA has the anticodon that corresponds with the mRNA. Now, these two don't go together without ribosomal RNA. Okay, ribosomal RNA, if you go back to the previous video, this is like the workers. These are like our construction workers that put all of the building material together. So let's take a look at how this actually works out. So our ribosomal RNA, our ribosomal complex, will come in and attach to the messenger RNA. Now, something to understand about ribosomes. Ribosomes are actually made out of various proteins put together. It's called a ribosomal complex. But for our purposes, we'll just think of it as one particular organelle. But where it does come in handy to understand it as a complex is right here, where you look at the three different sites of where protein synthesis occurs. You kind of look at it as a reverse word ape, okay? EPA, ape. So the first tRNA is called amino-acyl tRNA, and then the second is the P is peptidyl tRNA and decylated tRNA. Now, you don't actually have to memorize these technical terms. In fact, I've hopefully made it a little bit simpler for you in thinking of it this way. Your A site, think of it as your acceptor site. This is the site where the tRNA is accepted, and then your P site is your peptide chain. This is where the peptide chain is put together. So as protein synthesis occurs, you're always going to see the peptide chain grow off of the P site. And then you have your E site, your exit site. This is when the tRNA goes, it's like an assembly line. It goes to the A site, P site. P site is where it drops off the peptide, okay, the amino acid. And the E site is where the tRNA will eventually exit, okay? Kind of leaves out the back door, if you will. All right, so let's take a look at how all these three components come together and produce proteins. All right, so here again I have my ribosomal RNA and my messenger RNA. So if we take a look at the A site, the acceptor site, you have a codon there that's in place. You have a GAU codon. So we're going to take a look at our chart. This chart coincides with the codon on the messenger RNA. So you have a GAU. Okay, so we have our G's, and we narrow it down to our second base, A, and then our third base, U, it narrows it down to aspartate. So then we bring down our amino acid, okay, or our tRNA that contains the amino acid and the corresponding anticodon, right? And we have now our first amino acid in the peptide chain, okay, in our amino acid chain, okay? So now your tRNA will move down from the acceptor site to the P site, right? Your processing site, you can say. And then we move on to the next one, the codon. So the codon for CUG. So C, and then our second base, U, and then G. That codes for leucine, okay? So then we bring in the next tRNA, and then we make a peptide bond. That yellow little dot that you see, that's called a peptide bond. So we're making a peptide chain and amino acid chain. I know these have a lot of terms, so it's a lot of terms for the same thing. So I'm saying the same thing the whole time, just using different technical terms. So we're making a peptide chain at our P site. So you're going to notice the tRNA on the left move over to the exit, okay? And now we have, you'll notice that our amino acid chain stays on the P site. So the next codon, GAG, will code for glutamate. Okay, glutamate will then make a peptide bond with leucine, and then it'll just move down along the line. And then our next codon, AUG, methionine. Okay, so methionine will then come in at the A site, and you'll notice that the previous our tRNA right here is going to leave from the exit site, okay? And then our next amino acid, alanine. Alanine will now come in and make a peptide bond, okay? And we keep moving down the line, and this process continues until the entire mRNA code that is necessary for that particular protein is finished. Okay, so this amino acid chain will just keep getting longer and longer and longer, okay? And it gets long enough where it when the code finally finishes, and then it will fold. It will fold into a stable form, and then we have a functional protein. Okay, so at this point right here, we still don't have a protein. We just have an amino acid chain. And now, when you have the very specific bonds between these nucleic bases, between these amino acids, then we have a functional protein. So for example, this could very well be a protein RNA polymerase, okay? So then after it's been produced, the RNA polymerase or whatever enzyme it was being made will go now into the body and begin the process of doing whatever it does. Krebs cycle, electron transport chain, a protein embedded in the phospholipid bilayer. It can be anything. Okay, so just in summary, let's take a look at the whole picture now. So we look at DNA as the double-stranded helix, and this is in the nucleus. So the DNA molecule is too large to leave, so it transforms or it transcribes into a mRNA molecule. mRNA molecule will then go into the cytoplasm and meet up with ribosomal RNA and tRNA. Okay, and when these three components combine together, protein synthesis occurs and we make proteins. Okay, so that's RNA to protein. This is called translation. Okay, good luck in your studying.